Okay, so let's now look at another mechanism of action and in general another target for antibiotics. So that will be transcription and translation. And although this process has been very highly conserved throughout all the realms of life, and so there are not that many differences between prokaryotes and eukaryotes, there are enough that we can develop specific antibiotics. And especially, antibiotics, remember, have been made by other bacteria and trying to engage in a chemical warfare in order to control the environment around given bacteria. So let's go through. First, we have transcription of DNA to messenger RNA, and then RNA, the initial RNA transcript makes either a messenger RNA proper, the ribosomal RNA, and tRNA. At this point, there's addition of proteins to RNA, and so we form more important and more evolved machinery, which is the ribosome, which is a complex of proteins and RNA, and this has the overall task of translating mRNA, messenger mRNA, into a protein. And we do that, as you remember, by binding tRNA. First, tRNA binds to a specific amino acid, then it gets into the ribosome and recognizes the site where it needs to be added, and then the ribosome basically links together the new tRNA with the nascent peptide and ends up basically making our protein. All right, so with that, what can we do in bacteria to inhibit that process to our advantage? So we can have different mechanisms. So aminoglycosides, which is some of the amino acids we'll cover, act by binding to the 30S unit and basically cause misreading of mRNA. And so we have, nonsense protein. Chloramphenicol basically blocks the formation of the peptide bond. Other antibiotics bind at different sites and so interfere with assembly of the ribosome and initiation of protein synthesis. Tetracyclines bind again to the tRNA binding portion of the 30S, and so no protein is made. And erythromycin basically binds so strongly to the complex that prevents it from translocating and therefore to reading more messenger RNA. In general terms, the ribosome is comprised by a small subunit and a large subunit, and then that forms the complete 70S ribosome that was found in bacteria, which is slightly different from what we can find in eukaryotes. And so here is the structure of the ribosome, highlighting its complexity, and also in purple we have protein, in yellow, purple, and here orange and green we have RNA. So again, highlighting the complexity of the structure that comprises both RNA and proteins. So let's look at the first of the drugs we look at. Those are the aminoglycosides. So they bind, as we saw, to the 30S subunit. They prevent proper reading of the messenger RNA. They have a very broad spectrum of activity. They are made by Streptomyces gryzus, and the most famous of them are gentamicin and streptomycin. So what we need for activity is that the amines that form the structure of the of these antibiotics needs to be protonated. And so one of the mechanisms for resistance we'll see is to simply chemically modify them. They're not well absorbed per us. So this is a streptomycin, and again, the main resistance is obtained by protonating or phosphorylating rather. So these groups needs to be protonated. So phosphorylating or acetylation of these groups prevents its activity. Streptomycin is known to have important side effects at the level of the kidney and also to to cause basically side effects on hearing, and so to damage nerves in charge of hearing. Gentamicin is also an aminoglycoside, and so the same thing is true. So we have basically this is a aminoglycoside. So we have glycan linkage between glycosides and which have been modified to contain amines. These amines needs to be protonated for action, and again resistance is given by modification of these free amines. All right, so let's look at another class of antibiotics you're probably very familiar with, and those are tetracyclines. Tetracyclines basically prevent the binding of the tRNA, which has been already modified with an amino acid to the 30S subunit, and they are taken up by the bacterial cells by active uptake. So there is a specialized transport that they exploit. And the reason why mammalian cell resistance is because the ribosomal RNA is a little different, so we have a different binding. But most importantly is because eukaryotes don't have the transport that actively takes it in. Now, binding to the site of action requires the presence of magnesium, and because this class of antibiotics bind very strongly to divalent cation, they can bind to calcium and interfere with the formation of bones and of dental enamel. And so when they were first used, for example, a lot of children that were exposed to that will grow up to have stained enamel in the teeth, and so that's obviously an aesthetic problem, but it's still very serious. One of the things is because this is fluorescent, it can actually be observed in bone tissue basically for years after it has been taken. And the half-life correspondingly is pretty high, it's between 7 and 20 hours. So these compounds also tend to be not very soluble. On the other hand, they have a broad spectrum of action and they're generally well tolerated. So we talked about the most severe of the side effects. So let's look at the general structure. So they have these 4 fused rings, right? One of which is aromatic. And they're obtained by fermentation, again, from Streptomyces. They are also obtained by semisynthesis. We have 8 of those that are used in the market, which are all natural products. And in general, what we see is that what we can change is only the top part of the molecule, so only this part, right? And so the groups R1, R2, R3, and R4 can be exchanged in these related antibiotics. So let's look at their acid-basis properties. They are important because they interfere with their solubility. And so we have 3 pKa's that we can observe for this class of antibiotics. The first one is so-called pKa1, which is the most acidic, and is obtained by deprotonation of this alcohol. And so the reason this pH is so depressed compared to what we always see for alcohols is the presence of resonance forms. And so I'm drawing a simplified version here of tetracyclines, right? And so let's see what happens when this alcohol is deprotonated. then we have that the negative charge can be in resonance with pretty much everything else in a molecule. So on one side with the carbonyl, and on the other side with this carbonyl group. And so the high delocalization of the charge is responsible for the change in pKa, and the fact that the pKa is so much more acidic than what we would expect for an alcohol. Similar phenomena are observed for pKa2, where also we don't know exactly which one of the two OHs get deprotonated, probably this one or this one. And again, this stabilization is by resonance. We can see that here, this double bond is conjugated with the carbonyl and in turn conjugated with the aromatic ring. And the third one is the, kind of plain vanilla pKa that we observe for a tertiary amine. Okay, so because of their complexity, tetracycline can form several epimers, and so this epimerization reaction happens in the presence of acids or bases. And that interferes with activity because the quatrimycin is a lot less active than the natural ones. The degradation correspondingly is basically carried out either in acids or bases, and in both cases results in a ring opening here in a base situation. And this is then responsible for the loss of activity. In this case, in an acid, we have the formation of another aromatic ring. All right, let's look at another class of antibiotics called the macrolides. Erythromycin is one of them. So they tend to be active on gram-positives. They bind to the larger subunit of the ribosome, and they basically contain this 14-member lactone ring. And lactone ring, remember, is a cyclic ether, ester, right here. And then this nucleus is then glycosylated. So what they do by binding to the large unit, they basically inhibit the elongation of the protein, and prevents also translocation of the ribosome from one side to the other. And the exact mechanism is not totally clear. Most likely it actually does both. So they're bacteriostatic for most bacteria, meaning that prevents the growth, but they also can kill a few of the gram-positive ones. Okay. Another one is another antibiotic is rifampin, is a erythromycin, which targets the prokaryotic, basically DNA primed RNA polymerase, and so basically blocks the formation of the first phosphodiester bond when forming RNA from a DNA template. It's used mainly actually for tuberculosis. It's also used to resistant bacteria, and it's a very complicated molecule. Once again, that is not very available per us. The last one that we'll cover is chloramphenicol, which also targets the large unit and inhibits the transfer of the peptide in the formation of the new peptide bond. It works because it kind of resembles a peptide, and so kind of mimics that unit. So what else? Is selective for the bacterial ribosome and not for the eukaryotic one. So the toxicity of this antibiotic is related to an interesting phenomenon. If you think about it, what has ribosomes that resemble bacterial one in eukaryotic cells? And that will be mitochondria. So mitochondria, have been speculated to originate from prokaryotic cells, and they contain 70S ribosomes instead of 80S ribosomes. And so they are basically they can be inhibited by some protein synthesis inhibitors, including chloramphenicol, and that basically explains their toxicity at the mitochondrial level. Um, because of that, the kind of cells that are most likely to be inhibited, the eukaryotic cells, so off-target action, are those that have mitochondria which are highly active, and so those will be cells that undergo rapid growth and multiplication. Okay, so here's a little summary of antibiotics and their site of action, which should be pretty self-explanatory. We have looked at many of these compounds, although not necessarily all of them. As you can see, we have focused on a few because of their importance in a clinic, but also because we understand the mechanism of action relatively well. And the question that most of you will have after such a complicated chapter with a lot of material is what do we need to know? So what do we need to know about antibiotics? I would like you to be able to define the mechanism of action for the main classes, and that includes basically what we covered in the lecture. So the beta-lactams, the macros, the macrolytic ones, and all the antibiotics that act on the cell wall formation. The other thing that I would like you to remember is basically problems associated with antibiotics, especially with the beta-lactam antibiotics. So the mechanism of action that involves attack of a nucleophile on beta-lactam ring, both in terms of mechanism of action, but also in terms of beta-lactamases, which now destroy the amino acid. So you should be able to pretty much recognize anything, and be able to assign classes of antibiotics to a given molecule, and that's true especially for beta-lactam antibiotics. We have done a little bit of QSAR, not much, but we have talked about things that improve the activity or prevent problems, especially with the beta-lactam antibiotics. And then I'd like you to be able to discuss, the effect of pKa, basically with sulfa drugs and tetracyclines.